Welcome to today's lecture on cell biology, focusing specifically on microorganisms. In this lecture, we will explore the fascinating world of microorganisms and delve into various topics such as their basic structure, cell division, energy metabolism, interactions among microorganisms, antibiotic resistance, viral infections, biofilms and chronic infections, microbial motility, signaling pathways, host microbe interactions, virulence mechanisms, and advances in microbial genomics. Microorganisms, also known as microbes, are tiny living organisms that can only be seen with the help of a microscope. They include bacteria, archaea, fungi, protozoa, and viruses. Each microorganism possesses its own unique structures and functions. Bacteria and archaea have a simple cellular structure, consisting of a cell membrane, cytoplasm, and a genetic material known as DNA. Fungi have a more complex structure, including a nucleus, cell membrane, and cell wall. Protozoa, on the other hand, are single-celled eukaryotes with specialized organelles. Viruses, although not considered living organisms, contain genetic material encapsulated in a protein coat. Cell division is a vital process for all living organisms, including microorganisms. Bacteria and archaea primarily reproduce through a process called binary fission, where a single cell divides into two identical daughter cells. This process involves DNA replication, followed by segregation of duplicated chromosomes and formation of a septum that divides the cell into two. Fungi, protozoa, and viruses employ different mechanisms for reproduction and replication, depending on their specific characteristics. Microorganisms exhibit diverse metabolic strategies to obtain energy for their survival and growth. Bacteria, archaea, and fungi can be classified based on their energy sources, including phototrophs, using light energy, chemotrophs, using chemical energy, and heterotrophs, obtaining energy from organic compounds. Some microorganisms can carry out photosynthesis, converting sunlight into chemical energy. Others obtain energy by breaking down organic matter or engaging in symbiotic relationships with other organisms. Microorganisms often interact and communicate with each other through complex signaling pathways. These interactions can take various forms, such as competition for resources, cooperation in biofilm formation, or pathogenicity towards hosts. Quorum sensing is a common mechanism used by bacteria to orchestrate collective behaviors in response to population density, allowing them to synchronize gene expression and coordinate activities. The emergence of antibiotic resistance among microorganisms is a significant public health concern. Due to the overuse and misuse of antibiotics, some microorganisms have developed mechanisms to evade or neutralize the effects of these drugs. This resistance often arises from genetic mutations or the acquisition of resistance genes through horizontal gene transfer. Understanding the mechanisms of antibiotic resistance is crucial in developing strategies to combat this global challenge. Viruses are obligate intracellular parasites that infect host cells and hijack their cellular machinery for replication. They can infect various organisms, including humans, animals, plants, and bacteria. Viral infections can cause a range of diseases, from mild flu-like symptoms to severe illnesses such as Ebola or COVID-19. Understanding the viral life cycle and host-virus interactions is fundamental in developing effective antiviral therapies and vaccines. Biofilms are complex microbial communities that form on surfaces, such as medical devices or natural environments. Microorganisms in biofilms show increased resistance to antibiotics and host immune responses, making them challenging to treat. Chronic infections, such as dental plaque or urinary tract infections, are often associated with biofilm formation. Research in biofilm biology aims to understand their formation, quorum sensing, and mechanisms to develop strategies for prevention and treatment. Microbes exhibit a variety of motility strategies to navigate their environment. Bacteria and certain protozoa use flagella, cilia, or pili to move in response to environmental cues. Some motile microorganisms can perform chemotaxis, directed movement towards attractants or away from repellents. Understanding the mechanisms of microbial motility allows us to comprehend how microorganisms colonize different environments and interact with their surroundings. Microorganisms employ intricate signaling pathways to regulate gene expression, adapt to changing environments, and coordinate cellular responses. These pathways involve the transmission of signals through receptors, sensors, and intracellular signaling molecules. Examples include two-component systems, cyclic AMP, CAMP, signaling, and quorum sensing. Elucidating these signaling mechanisms provides insights into microorganism behavior and can inspire the development of therapies targeting specific pathways. Microorganisms can exist as commensals or pathogens within their host organisms. Commensals establish a symbiotic relationship with the host, often providing benefits without causing harm. Pathogens, on the other hand, can cause infectious diseases by evading host defenses and interfering with cellular processes. Studying host microbe interactions helps us understand how microorganisms colonize, persist, and interact with the human body. Virulence refers to the degree of pathogenicity of a microbe, determining its ability to cause disease in a host. Microorganisms employ various mechanisms to establish infections, evade host immune responses, and cause damage to host tissues. These mechanisms include adhesion to host cells, secretion of toxins, manipulation of host immune responses, and invasion of host tissues. Unraveling the intricacies of virulence mechanisms aids in developing targeted therapies and vaccines against pathogens. Recent advancements in DNA sequencing and bioinformatics have revolutionized the field of microbial genomics. The study of microbial genomes provides valuable insights into their evolutionary history, metabolic capabilities, and potential virulence factors. Comparative genomics allows researchers to identify genes responsible for specific traits, track the spread of antibiotic resistance genes, and understand the genetic basis of microbial diversity. These advancements have opened new avenues for diagnosis, treatment, and prevention of microbial infections. In conclusion, the study of microorganisms is crucial for understanding their structures, cellular processes, and impact on health and the environment. Exploring topics like genomics and infectious diseases helps us develop strategies for disease prevention and treatment, advancing medicine and biotechnology.